that eukaryote complexity itself cannot have derived from a proportional increase of protein coding genes. At first, people thought we would find at least 300,000 genes because humans are complex beings. In the human genome, it turned out to be only 25,000. So what then is explaining this complexity? And one of these ways of looking at it is this concept of causal emergence, wherein increasingly in eukaryotes, there is, it's the interaction between the molecules that becomes more complex. And this is a little snippet from this paper here, wherein it's shown that in eukaryotes, there is a lot more interactions between proteins than there is in prokaryotes. And it makes a ton of sense, right? So instead of making more proteins, there is exponentially more connections between the proteins to be explored than there is new proteins to be invented. And therefore, we were wondering if this is the case, could it be that the eukaryote complexity derives from the exploring of the combinatorial space of biomolecular interactions, which is obviously orders of magnitude larger and probably easier to access evolutionarily? And if so, could we directly measure it in an unbiased way? Yeah, obviously, we think yes. The answer is yes. That is by measuring the edges instead of the nodes in a biomolecular network of the phenotype. And this is what this paper is all about. Now, how did we measure edges? How do we define these edges? I'll come to in a minute. I first just want to take you on a very small detour on why it was histones, metabolome, and proteome that we decided to measure in this multi-omics approach. And therefore, I'm going back to the first eukaryotes. They arose from a merger between a bacteria and an archaea. The bacteria there became the mitochondria, the mitochondrial endosymbiosis, and actually a bit later on in the evolution of eukaryotes, it happened a second time when chloroplasts were engulfed, giving rise to plants. So I think it's fair to say that the prokaryotes, the bacteria donated energy to the eukaryote experiment. But what was it then that the archaea were donating? The Ascard archaeota, to be exact. The only thing we do know is that genome size in eukaryotes exploded and they invented multicellularity. And in a way, these are two sides of the same coin, the coin of information. Because think about it, genome size is information content and multicellularity is information control. So if you look at the size of the genomes, eukaryotes don't even have an overlap in size with their ancestors on a log scale. And eukaryotes have up to a 200,000-fold as complex genome as the prokaryotes, as the bacteria and the archaea do. And from an energy point of view, that makes sense because replication only takes up about 3% of the energy budget of a cell. Yet this huge increase in the genome is not reflected in the proteome, as only 1.5%, for example, the human genome codes for proteins, making it so that the proteome, the set of all proteins, is only up to 50-fold as complex as that of archaea and bacteria. And again, from an energy point of view, that makes sense because translation is very expensive. It takes up about 75% of the energy. But also you need to contain all this information. You need to make sure that it's always read out in the right way. For example, when in multicellularity, when an embryo develops into a full organism, it's very important to have the expression profile of the different proteins always very finely tuned throughout development, obviously, but also internally differentiated tissues, because expressing their own protein can lead to cancer. And we strongly believe, obviously, and others, we have this is kind of a flow that is arising in molecular biology, is that histones are the one that can mediate these two different aspects through their well-known role in epigenetics, because obviously it's the histones that roll up the DNA into the nucleus, but it's also together with DNA modifications. It's the histone PTMs that decide what part of the genome should be accessible for transcription and translation. And if you then return to this eukaryote experiment, again, the energy deriving from the ancient bacterium and the archaea donating the histones is what we think now. And we can still probably see this because the histone RNA has no poly A tail. People have started suggesting that the entire metabolism effectively can be projected on top of histones as PTMs. So they covalently bind to the histones and become PTMs for the cell to read out the energy state of the cell. And this is why every year still new PTMs are being discovered. Histone lactylation, succinylation, benzoylation, crotylation, and it goes on and on and on. Even so far as to find serotinylation and dopaminylation that mediate cocaine addiction in a rat brain. And if you think about it, this is a very efficient way to store the information. You can come up with up to a Google different proteoforms if you look at all the different combinations that can be built between these PTMs. And this has led us in our lab to kind of redefine a bit how we look at molecular biology, if you will. So the basic view obviously is that the genes have the information and it's transcribed into RNA. These become proteins and they metabolize the metabolites. So you go from an information towards an energy flow. So the organism is trying to harvest the energy to live. But if you insert the histones and epigenetics in general with DNA methylation there as well, you effectively insert a pivot point, allowing this flow to fall back onto itself, whereby these metabolites are projected onto the histones, and this tells the cell what is available or what is not, so that the cell can express other proteins to harvest new sources of energy. And this could be a perfect circular continuity, a way for an organism to adapt to its environment. We started calling this novel edge, if you will, in this very iconic small network here, the energy information axis, because it translates what the energy is in a cell and how it's turned into information by putting it onto the histones. And we feel like this interconnectivity starts to kind of support and maybe even it introduces essentially this basic level of causal emergence, if we think about it. So this is how we look at it right now. And this is also how we came to this manuscript. It's still before submission when this is being recorded. I just wanted to show you what it is we've done. We had 18 AML cell lines, 6 replicates each. And sequentially, we've extracted the proteins, the metabolites, and the histones from each of these pellets with a very simplistic protocol. We crash the proteins in acid, and the supernatants is crashed again to get the histones into the pellet, and then we have the metabolites in the supernatant. So we had well over 100 proteomes that we measured in a single batch, 100 histone samples, which we measured in one big batch, and then the metabolites were measured in different batches, as is often done in metabolomics, on C18, on Hillig, positive and negative mode, but there's also 100 metabolomes, if you will. And we integrated these through 2 main frameworks. One is in-house developed, the Lemonite or Lemonite workflow, and the other is through Bigomics, which is a commercial software that allows you to integrate such things. And this is then how we present this reasoning in the manuscript. We add a little level here, which is the directionality of the circular graph, whereby we show what I think the bulk of the attention has been about, which is sequencing-based technologies, DNA methylation, ChIP-seq, RNA expression, finding mutations in cancer, looking at post-transcriptional modifications. But we are now looking at the histones, the proteins, and the metabolites. And we've added these kind of directionality arrows here to help you interpret what you could potentially conclude from the data, because we do not measure it. We do not measure these arrows. We measure the correlations between and within omics layers. And it's just to help you see what it could mean, which is, for example, if a certain PTM opens up the chromatin and produces certain proteins, this is the histone protein or HP axis. But then there's the writers and erasers, these proteins like PRC2 complex that kind of writes a certain PTM, and then you have the pH axis or directionality here. The proteins themselves, they convert metabolites, PM axis, but the metabolites are crucial to mediate the activation or degradation of proteins. So there's the MP axis, and then there's the metabolites being projected onto the histones, which we call the energy information axis or the MH axis here. And we were not aware of an inverse directionality whereby histones would donate a metabolite before we started mining the data, but that's indeed what we found in the end. So it helped us to effectively look for signs of this kind of HM interaction as well. So what's new? What is it that we're trying to do? What is new? What are we bringing to the table? First of all, this MS-based multi-omics workflow of the molecular phenotype is, according to our knowledge, new. Nobody's done this from a single cell palette. So these are very truly entangled omics layers, which we can manage to extract from a single cell palette and measure them with a mask. This kind of reasoning, so the actual circular path graph that I'm showing here, I think is also, it's not new, but it hopefully will help in the future to facilitate discussions. The fact that the importance of the edges in the network is essentially what we're highlighting and how we are approaching the data at this stage. And by doing so, by iteratively looking at this data, because it's so extremely rich, there is up to 30 million edges in this data. So we never intended to find a given biology from this and then do a mechanistic follow-up. This is the data for its own right. It's just worth looking into. And while we were mining it, we kind of found different ways in which molecules were showing coordinated behavior, be it positively correlating or anti-correlating. And we now have this overarching term, functional entanglement. The first level is very obvious. Turns out that you can very easily find protein complexes in this data by just ranking Pearson correlations between proteins, and the most correlating proteins are very often those that belong to the same protein complex. We didn't know upfront, but this is what the data is telling us. So you can do quite some interactomics data mining without ever being there ever being a bio-ID approach or alike. Then there is these known functional co-regulations. For example, if you look at H3K27 trimethylation, the most strongly correlating protein out of 4,500 is EED, right? Which is a PRC2 complex protein. So this is telling us like the EED was the one writing this PTM. So kind of, you would say obvious, but it's there. I mean, you could just read it off the data. So these are the ones that actually tell us how valuable the data is. The more of these we find that are already known, the more we know that this data is effectively very noiseless and very valuable. Because then there is these nth order functional entanglements where we don't know what the link is anymore. But it's about first defining that the link is there, showing that over 100 cells, different cell palettes, give you a very strong correlation with a molecule you've never linked to one another before. That tells you something, because all the other molecules that correlate very strongly to it, they are known protein complexes or known functional connections. So it's about finding these new members in this list of correlations. We also try to introduce the concept of this energy information axis. Again, the concept is not new, but maybe the name at least sounds to us very applicable. But the thing is that this manuscript is all about the data itself. It's about this data set, which is too big for one group to handle. So this is a big shout out for others to go and look into these 3 different reports that we have generated, looking for their own targets. There is very many to find here. So this is now where I will start giving a bit of a tutorial on how to approach the data. And I'll use some of the things that we describe in the manuscript, but as you can imagine, you can just start filling out different names and you will see other biology. So voila, this is how that looks like if you arrive on the platform. Probably I was logged out already, so yes, and the password will be gone by the time that we publish this. So you will enter here in this Lemonite web page that was created by Boris van der Moortelen from the lab of Vanessa Vermeersen. And this is where everything is bundled. So you have one link, you click it, and you can access everything from here. First thing you can do is to download this interactive network. If you do so, you get a bit of an overview of everything inside the data. And obviously that is a big hairball, right? A big hairball plot. This is how that looks like, but it's interactive, it's browsable, and you can look for your favorite biomolecular target. Maybe when I go back, I can show you that everything we've detected is in these 3 text files. If you download one, this is how the hPTMs look like. These are all the PTMs that are in the data. You find your favorite one, say H3K27 trimethylation. And voila, this is how it's named. This is how the nomenclature we used. This is the network. You can then just look for it, click it, and it's highlighted. So how is this network built? It's essentially protein modules. These are proteins that move in the same way throughout the experimental design, in this case, throughout our 18 different AML cell lines. And you can see pop-ups that show what are the regulators, as we call them, which are effectively the hPTMs and the metabolites that are in the network. We call them, for example, here, N6 and 6 and 6 trimethyl lysine, which is the metabolite that arises after a protein with a trimethylation is being degraded. And this in its own is a precursor for carnitin biosynthesis, for example, acetylcarnitin. And you can see how we are already finding some common grounds here, right? So things are starting to be linked. We have positive edges, negative edges. Again, we use the concept of a regulator here as a bioinformatics term, not as a biology term. It does not imply that this given metabolite, for example, is inducing this module for some reason. We are just saying they move the same. They have this edge between them. And so there seems to be reason or something to believe that they are together. As you can see, these 3 metabolites already, they all link to this module number 16. And from the pop-up, you can see how we already have some gene set enrichment analyses there. But at this point, it's probably more interesting to start searching for the things you're interested in. Let me, for example, again, look for the PTM and search for all the modules that this PTM connects to, and you can see them all listed here. The more you have, the more densely connected this biomolecular feature is within the network. So this is then what this derives from. So to the right, you can see how this data looked like. All the samples, the 100 samples, are here in the bottom. And you can see this is module number 16, bringing together all these proteins that go from this blue to that yellow. So they are, by the algorithm, considered to be part of the same module. And then there is at least 4 PTMs and 6 metabolites that were appointed to this module, if you will, as a regulator. The main one here being H3K27 trimethylation, which is positively correlating to the module, and the first metabolite here also positively correlating despite this one cell line here is trimethylation. So already you can see how these things are connected. And if you want to know what module 16 is all about, here is your gene set and enrichment analysis. So just to be clear, this network was built using only about half the proteins that were identified, being the ones that are the most variable proteins. So the beauty of the network is you have instant access to these modules with their gene set enrichments, knowing how these things are linked, but not all the data is inside at this point. That said, I think what's very striking is that we noticed how a lot of these modules contained known protein complexes, right? So this is what this orange bar here is. These are the quorum database protein complexes that are known. They are plotted automatically. If there is certain proteins that are known to be part of the same complex, it should show up as these orange little squares. And in this case, it's the nuclear pore. So these NUP proteins are all part of the nuclear pore. So essentially what this is showing you is that there is apparently a link between H3K27 trimethylation and the nuclear pore, according to this one edge. I'm not saying this is already biology, but this is what it's telling you. And I wasn't aware before, but turns out to be a thing. Effectively, PRC2 is connecting or can bind to the nuclear pores, forming one big complex. So there seems to be a known link. So these histone PTMs here, not many people measure them with a mass spec. And so we thought it's very important to give a very elaborate report on how we quantify a single histone PTM. And you can download this HTML right here. And if you open it, this is how it looks like. Voila. This is a script that is based on MSQROP to PTM, developed by Nina de Meulemeester from the lab of Lieven Clement. And it was tweaked for histones by Ruben Albay from our lab. And it walks you through the different steps that we had to take to quantify these different histone PTMs, the ones that were searched. So if there's other PTMs that you want to search for, this is all possible. In that case, you would have to go back to the Progenesis project, which is also publicly available. And we have a full manuscript that describes how you can handle Progenesis to kind of look for your own histone PTMs. But so, yes, we show how we align the different variants of the different histones. We show the extraction efficiency, quality, QC plots, if you will, normalization plots, how many peptidoforms for each of the histones we found. And then here is a dimensionality reduction plot that you can color code according to the different metadata. This is all the metadata that we have present in our data set in these different AML cell lines. Here we show whether your nucleosomes are consistent in shape, if you will, in stoichiometry, which seems to be the case in this. So that allows us to do a variant agnostic workflow. I won't go into too much detail. And then we can start inferring histone variants, PTMs. We even have some co-extracted proteins there. We call them acid extractome. And all this inference use happens through the Q features from Bioconductor from Laurent Gatteau. And then we end up seeing single PTMs. We can define our contrasts, well, our cell lines first. Here is all the contrasts that were defined. And this is then how that looks like. So here we have, for example, the meto high, meto low phenotype defined by Matthias Mann and others recently. You can just mouse over and it shows you all the variants that we cannot distinguish and the PTM where it sits. If you are interested in what we've measured, then you have the peptidoforms. These are somewhat complex for people not used to looking at this data, but it is from these peptidoforms that we infer the abundance of single PTMs. And for example, CD34, which is a very known biomarker in AML, you can see here it has these H4K12 acetylation, H4K8 acetylation, K16 acetylation. So there's a clear enrichment here of the H4 end tail. You can look at how we came to these quantities by downloading the figures and the essays. And this gives you a bit of a feel of what we are up to, because if you want to quantify H3K27 trimethylation, for example, here we have 4 peptidoforms that all cover this PTM. So we have to sum these 4 in order to get an abundance of this one PTM. Sometimes there is many more. And this is exactly what is depicted here. So we have the abundances in the top as they were measured. And in blue, we see the protein abundance of H3.1. And these two are then used for normalization. And this is what you see below here. And then all these abundances are summed into this thin red line. And then the software adds the straight, thick pink line as the model for this PTM. So this is all how we came to the histones, and you have an interactive report to go and browse it. You can define your own contrasts here. You can see how the contrasts were defined. So there is still some wiggle room here to play around. And then going back to the central hub, which is this Lemonite website here, the final thing you can do is also go to the Bigomics Playground. If you click it, this is a commercial software, so you end up in a different website. If you register, you get access to the platform. And I was here just now refreshed. So my browser refreshed. So that means I'll probably have to log in again. This is exactly how that looks like. You would get an email to verify that effectively you're trying to log in. And with this login code, voila, we should be able to enter. Okay. Now, if you register for Bigomics, you can go to public datasets and you will find our 2 datasets there. This is the multi-omics dataset. It says multi-omics. And the other one here is called AML 18 cell lines, June 2025. And you will have full access to the platform as I am showing it right here. So I am now opening the Proteomics platform, but we also have the multi-omics dataset. So these are 2 different data sets that you can play around with. And there's obviously the normal plots, the conventional QC plots, some t-SNEs, some clusterings. You can see how the contrasts that we've defined and the data that is entered. You can look at your samples, how they cluster, for example, or what proteins are most differential throughout the workflow. And for example, here is CD34, so it's among clearly the most impacted proteins in this data set. For example, we can also just correlate our cell lines to one another. They cluster very nicely. And there's little reason for me to go through all these functionalities. What I want to point out here is that people tend to do these binary comparisons between two phenotypes, if you will. This one now is showing you 10% FBS or 20% in the FBS in the medium already. It's quite shocking how big an impact this has. What the underlying biology is at this stage, I wouldn't go into, but if we do, for example, CD34, people would look at these volcanoes. Everybody does. It's very useful, and you can see all the upregulated proteins in CD34, all the downregulated proteins in CD34, amongst which is, for example, asparagine synthase, which is described in the manuscript. But the thing is, if you ask for biomarkers in this feature from Bigomics, it will effectively try to calculate what is the contribution of each of the proteins when I try to cluster my cells in CD34 positive and CD34 negative. Obviously, you would expect CD34 to be the first hit, right? But it's not. It's here. Every iteration, it will be somewhere else because it's machine learning. It does all these different random forest, XGBoost, correlations, regressions to try and cluster your cells. And each of them is scored, and then they compile the different scores one onto the other. But the point is that CD34 here, this is the phenotype I asked for, has at least 10 to 12 or 15 other proteins that seem to be better biomarkers for the very biomarker I asked for. And that is not so surprising if you think about it, because this is actually the tab where we can mine the edges. Okay, so within the proteomics, this is now no longer multi-omics, but I can simply ask to show me CD34. How is it expressed over these more than 100 samples, or just a little below that are in Bigomics? And this is how that looks like. So every time CD34 goes up, this protein goes up with it, which is ITGA6. And while these two apparently are known to be together in bone marrow cells, the third one here, so this is, you could say this is a non-interaction, or they are even part of the same protein complex, but this one is a short chain short chain acyl-CoA synthetase, which is a metabolic enzyme, which has no clear direct link with CD34. But if you look at the expression, you can see that irrespective of the genotype, whenever CD34 goes up, so does this enzyme. So it's only then that you can start looking into how these two are linked. Look at macro H2A. This histone here is tightly correlating, and these are very high correlations. If you look at it, it has 4,500 proteins in here, and these correlate. These are the highest correlating ones. On the other side of the spectrum, it's just as interesting. What is always going down whenever CD34 is going up? Well, for example, asparagine synthase, which from the perspective of the asparaginase therapy in ALL is a very fascinating biomarker here. And we know that in AML, the metabolism is going to shift drastically from the bone marrow to the periphery when they are starting to differentiate. But this is many of these proteins are directly and tightly correlated. Now, to give you an example of how you would be able to reconstruct protein complexes, let me take this PRC2 member, EZH2 again. So we're going back to H3K27 trimethylation. We're kind of moving back into the multi-omics here. And what you see, if I maybe just focus on the visual part here, this is again, a ranking of the most correlating proteins. While the number of this correlation might not feel very high, they are ranked, and there's 4,500 proteins here. The first one is CIS12, which is together with EZH2 and PRC2. And then there's EAD, also a member of this complex. Apparently, SMARCA, I didn't know, is also a member of this, or binds a lot to PRC2. And this one, BAS1B, we just last year found in a BioID study to also be complexed to PRC2. So you are reconstructing protein complexes right off the correlations, saying that these two in the middle very probably also are linked to PRC2. And it turns out ADNP is well known. We're not sure about SRRT, but that makes it that more interesting for mechanistic follow-ups. So we can, the highest correlating proteins, if I, for example, want to look at the nuclear pore, I can take one of the members here and see what that complex is with, and you can see how we reconstruct the nuclear pore right here. And then there's obviously these proteins in the middle that you wouldn't have expected there. Also, it's very important to see, you can verify what this correlation is, because the Pearson correlation is just a number. And for example, here, it's mainly driven by a single cell line. If I quickly go and see which one, we can just depict it like so. And then there's this brown dots here. This is MOLM16, is the one driving at least this correlation. So, but at least you can manually verify what is happening here. So obviously there is a lot of systems bio options here as well. WGCNA, it takes too long to render, but you probably know what it does. You can just look into this. Maybe that's to end off here. I can go to see the multi-omics setup, which is also available for you to browse. It has a few extra multi-omics features in the latest versions of Bigomics. But again, first of all, if you go to the correlation analysis, you can see how the histones all start by HX, the proteins by PX, and the metabolites by MX. But in terms of correlations, you can just start looking for, say, this trimethylation. Let me see if I type this. Here it is. This is a trimethylation of lysine 27. And the first protein in a list of now 5,500 entries, the first protein to correlate is EAT. So clearly the correlations will be higher within omics layers. But even between omics layers, we can see these things. So EAT is the writer of this PTM. And again, it's all the way on the top. So obviously, again, multi-omics is a bit more murky, but there's little use in looking at the absolute values. It's all about the ranking. Because if you think about it, you can now effectively rank all these Pearson correlations like you would do for fold changes in a binary comparison, and then do a gene set enrichment analysis. The thing is, that is also already pre-done for you at the central hub, which is the Lemonite webpage here. Because if you scroll down a bit, I always try to clear all inputs, and sometimes you have to push a button twice. These are minor things. But this is the second part here, functional entanglement in the full cellular context. What this does, and I can do it, for example, for EZH2, I hope. I haven't tried it. There's so many proteins, voila. And then you can see what it correlates to, heterochromatin, right? So it anti-correlates functionally too. So what happened is, this is AZH2, and all its Pearson correlations to the 4,500 other proteins was ranked And this is again a sigmoidal curve, like it would be for a fold change in a cellular comparison. And instead of fold changes, it's Pearson correlations that are being fed to a gene set enrichment analysis. So effectively, it finds the highest correlating proteins to be functionally coherent. In other words, we are not defining the function of this protein. We are saying what its molecular environment in this data set looks like, functionally speaking. So this, you can do this also for histones if you like, or for metabolites for that matter. For example, if I go back to my list of different metabolites here, I haven't done much for metabolites, so let's focus just for a second here on the metabolites. I have this M-methyladenine, which we describe in the manuscript. It's derived from mRNA that was methylated and then broken down, and then it becomes a metabolite. We detected it both on the C18 LC system and on the HILIC LC system. We keep them both. So they both have their own correlations. So if I, for example, want to know now, how do the proteins correlate to this metabolite? This is how that looks like. And then you see that this metabolite anti-correlates to the respiratory chain complex 1 and the mitochondrial large subunit. Or if you look at reactome, the nucleosome assembly seems to be positively correlating here. So you can do this manually, obviously. You can simply export this list, especially if it's not the multi-omics here, but this list in the protein list, you can export just like so. There's CSV, you download it, and you can put it into, for example, string, where it will show you all the different correlations. So to end off here with the multi-omics project, there's a few cool features left. Price collecting Steiner forest, I won't go into, but for example, here the lasagna plot is plotting these different omics layers, and it's depicting the edges between the omics layers as a lasagna. So the 3D version is clearly the one that looks very nice, but there is a 2D multipartite graph just as well. So you can see here, you can then see which connections we can draw, which proteomics proteins, which metabolites, which histones behave exactly the same. There is the MOFA, which is more commonly used in multi-omics. And there's a multi-omics WGCNA right here. So you can see in the MOFA here that the first 3 factors are mainly proteomics. Then there's like factor 3 and 4 are more about metabolomics, and factor 5 is about the histones. And you can always go and check if any of these factors is functionally enriched for something. So I guess there's also deep learning, but I guess this is about it.